I have been doing. You're recording now. Okay, so hi everyone. Thank you for this invitation. And my name is Ariane Nunes Alves. Uh, I'm from Brazil. I'm not German. Since 3 years ago, I am a junior group leader at the Technical University in Berlin, in Germany. I'm the head of the theoretical structural biology group, where we do modeling and simulation of protein ligand interactions in vitro and in vivo. We work with two main topics. One of them is ligand binding kinetics, and the other is crowded cell-like environments. So on the side of ligand binding kinetics, we use physics-based methods like molecular dynamic simulations and enhanced sampling, and also machine learning to predict time scales for protein ligand binding. And from the side of crowded cell-like environments, we use physics-based methods like molecular dynamic simulations or Brownian dynamic simulations to see how the cell environment affects biological phenomena like protein ligand binding or conformations of proteins. Okay, just a brief introduction. In drug design, it's very common to quantify protein ligand interactions by looking at thermodynamics of protein ligand binding. And in equilibrium conditions, we quantify thermodynamics by looking at the dissociation constant Kd or the binding free energy delta Gb, which are basically ways of measuring the affinity of the ligands for the protein. So here we have Kd, the dissociation constants, where P and L are the concentrations of the protein and ligands alone in equilibrium conditions, and PL is the concentration of the protein ligand complex. Instead of writing this, we can write the binding free energy, which is this constant, which is given by this equation, where Kd is here, and R is the gas constant, and T the temperature in Kelvin. And something important as well is that Kd and delta Gb are state functions, meaning that we can predict them only looking at the end states of the binding process. But what my lab works on, what we are interested in, is quantifying the kinetics of protein ligand binding, or the time scales for protein ligand binding. And this is special because rate constants like K-on and K-off, they are better descriptors of non-equilibrium conditions or non-equilibrium systems. And a very special class of non-equilibrium systems are us and our cells. So our cells are out of equilibrium. They're constantly taking glucose and oxygen from the environment and converting it to CO2 and energy. And then, therefore, in steady state conditions, we have these two constants here, K-on, the association rate constant, which is the rate of complex formation, and we have also K-off, the dissociation rate constant, which is the rate of complex dissociation, and this can sometimes be expressed as the residence time tau, which is just the inverse of K-off. So most of this lecture, I'll either be talking about K-off or tau, the residence time. So I hope you remember these two abbreviations here. And final thing I want to share is that Ko and Koff are not state functions. So different from thermodynamic properties, when we want to quantify Ko and Koff, we usually need to know the pathway used to move from one state to the other state. Okay, and then in drug design, binding kinetics became an emerging topic since 2006. So in 2006, Copeland, in this paper here, he proposed the idea of optimizing the protein drug residence time to make better drugs. And the reason for this is that kinetic rates, in some cases in the literature, were shown to be better correlated with in vivo efficacy. And if you think about it, it makes sense because the longer the tau is, it means that we have a longer protein ligand, longer duration for the protein ligand complex, and therefore we have longer physiological effects and less of target binding. And here we have an example of what I just told you. So here, this is an example coming from this paper at the British Journal of Pharmacology. So the authors were quantifying, they were looking at inhibitors of the adenosine A2A receptor. They were measuring the efficacy inside cells. So here in this axis here and here, we have the efficacy in relation to a known inhibitor. And here in this axis, we have the inhibition constant, an equilibrium thermodynamic parameter. And here we have the residence time. So we can see that the coefficients of determination, the R-square values, they are much higher when we compare the efficacy with the residence times. So it's closer to 1. Unbinding kinetics can also be meaningful for enzyme engineering. And this is the case, first of all, because a lot of messages from my research group, sorry. So first of all, binding kinetics is relevant in enzyme engineering because more than 60% of the enzymes have the active sites located in deep internal cavities. So this is a protection to the catalytic site because then it doesn't easily interact with things in the environment. And therefore, we need some specific tunnels or pathways for substrates to access this catalytic site. And this gives us then the opportunity to modify these tunnels or pathway to optimize either substrate binding or product release to have better enzymes with higher catalytic rates. So just one example from the literature, this example comes from this paper here at PNAS. The authors were looking at epoxide hydrolase. They did simulations to see substrate binding and product release in the Y-type enzyme. And what the authors realized is that product release in this case was basically obstructed by these two bulky residues here. And they wanted to have a higher performance for this enzyme when this enzyme was degrading these products here, alpha-naphthyl glycidyl ether, NGE. So what they did based on their simulations was to introduce mutations to alanine either in this position or in this position, you see here or here, to optimize product release. And then they tested the ideas. So here we have the catalytic rate for the Y-type enzyme and the catalytic rate for the mutant in position 145 or for the mutant in position 128. And we can see that we have a higher catalytic rate, meaning that the enzyme became more efficient when we optimize the liberation of the product. And when we talk about kinetic rates, we can either predict then using data-driven methods or physics-based methods. And on the side of data-driven methods, we can use machine learning regression methods. The advantage here is that the predictions, once we have a trained model, can take seconds to happen. But the problem here is that we need a lot of experimental data or kinetic rates to train our model. And on the other hand, we can also have physics-based methods, which I'll show you a little bit today. So we have molecular dynamic simulations, usually combined with enhanced sampling methods. And in this case, the advantage is that we do not need experimental data to make predictions, but the predictions can take days to happen depending on how long it will take us to run the simulations. And if you want to know more about both methods, I recently reviewed both groups of methods to predict kinetic rates in these two papers here. So my lab is very interested in predicting kinetic rates using both data-driven methods and physics-based methods. But what I will show you today is an application of machine learning not to predict kinetic rates, but to identify unbinding pathways in the simulations. We will see this soon. So this presentation is roughly divided in two parts. First of all, I'll talk about the data set that was used as basis for the machine learning work. And then in the second part, we'll go to the machine learning itself to identify unbinding pathways. So in this whole presentation, we will now talk about nickel-iron hydrogenases, and especially this nickel-iron hydrogenase from the Desulfovibrio fructosovorans, which is represented here. So broadly speaking, nickel-iron hydrogenases are a model system to understand hydrogen formation and cleavage. So nickel ion hydrogenases, they basically catalyze this reaction here. So they can either break or form molecular hydrogen. And they have a lot of applications in biotechnology because people are interested in using hydrogenases to generate molecular hydrogen in an efficient way. And molecular hydrogen, on the other hand, is considered the fuel of the future because when we burn it, we just generate water. We don't generate CO2. People are particularly interested in this hydrogenase from this organism, the Desulfovibrio fructosovorans, because it has a high catalytic rate in the generation of a molecular hydrogen. But this enzyme is also inhibited by CO and O2. So there are a lot of investigations about experimental and computational investigations to make this enzyme resistant to CO and O2. Okay, so talking a little bit from the experimental perspective now, the group of Professor Christopher Leger in Marseille, in France, they created a method published in PNAS called protein film voltammetry, where they could measure kinetic rates for CO and O2 binding and unbinding to and from this catalytic site in the nickel ion hydrogenase. And they used this method and they designed a lot of mutants. You can see the proposed mutations here to see if they could achieve nickel ion hydrogenases which were resistant to the inhibitor CO and O2. You can read the paper later, and what happened is they could not achieve nickel ion hydrogenases resistant to CO or O2, but what they could do is to find some mutants that had lower, that had a delayed K-on value for binding of the inhibitors to the catalytic site here. And based on crystal structures and also in simulations, different groups around the world were investigating also what could be the possible bottlenecks for gas diffusion inside these nickel ion hydrogenases. And two bottlenecks were proposed in the literature. The group of Professor Christopher Leger proposed that the main barrier for CO binding and unbinding were the residues in positions 74 and 122. And another group working with simulations proposed that the main barrier, the group of Jochen Blumberg proposed that the main barrier was actually this distance here combined with the distance between residue 74 and 476. Okay, and taking all this information into consideration, in the first part of the work, we use molecular dynamic simulations to investigate CO dissociation for the Y type and for all the mutants of nickel iron hydrogenase, and we wanted to test if there was a correlation between the candidate bottlenecks and the K-off values in the nickel iron hydrogenase. We also wanted to see if the mutations in nickel ion hydrogenase changed the pathway for CO unbinding. And the second question here was our main motivation later to develop the machine learning model that I will discuss with you later. And for this first part of the results, if you want to know more, you can check the paper published this year at JCIM. And final point I want to make is that in the experiments, we have CO forming a covalent bond with the catalytic site, and then there's bond rupture, and then CO leaves the interior part of the enzyme. But in our simulations, we were using a conventional force fields, therefore we don't have the covalent bonds between CO and the catalytic site. We already start with the CO no longer forming a covalent bonds with this catalytic site here. So we just see CO diffusing away from the interior of the enzyme. Okay, and this is basically the paper. So if you want to check the paper, yeah, it was published in JCIM in May this year, the first part. I don't know how familiar you are with molecular dynamic simulations. So are any of you familiar with this?
Doctor, we are, like I said before, we are a multidisciplinary group. You have computer science here and medics, mathematicians.
Uh-huh.
So even if some of us would have an idea, but some I think they don't.
Okay, I'll explain very briefly then. Okay, thank you
Thank you.
for the clarification. Yeah, so molecular dynamic simulation. So ideally, the ideal equation to observe the motions of atoms and electrons would be the Schrödinger equation, but this would be incredibly computationally demanding for to study a protein in a box of water like we do in my research group and other people do. So to investigate simulations, no, sorry, to investigate proteins, we usually use molecular dynamic simulations, which make some approximations, but it still provides us useful information. So the first approximation, we don't use the Schrödinger equation to propagate motion, but we use simply the Newton equation of motion. So A is the acceleration over the atom, F is the force over the atom, and M is the mass of the atom. And then at every time step, we are always calculating the acceleration to see the new positions of all the atoms, and therefore what we need is a way to compute the forces. And the forces, in turn, are computed by the force field, which is basically a description about the bonded and non-bonded interactions that the atoms can make with the environment. So for the bonded interactions, we use harmonic potentials to describe the bonds between two atoms and the angles between three atoms. And we also have this equation here to describe the torsions, the dihedrals formed between four atoms. And for non-bonded interactions, we have this equation here that describe, the Lennard-Jones equation, which describe the van der Waals interactions. And we also have this equation to describe the electrostatic interactions between two atoms. And with all these equations and these approximations, we can achieve in our simulations a time scale of nanoseconds or microseconds, depending on the resources we have and the size of the system. Okay, and with all this that I said, we still have a problem because experimentally, unbinding takes milliseconds or more to happen, while molecular dynamic simulations reach the microseconds time scale at best. So there are a lot of methods in the literature called, broadly speaking, enhanced sampling methods that helps us to bridge this difference in time scales and facilitate the observation of the phenomena we want to see. That in this case is protein-ligand unbinding. And the method I will show you in this, the method that we used in this work is tau-RAND, which basically facilitates the dissociation of the ligands by applying a force of constant magnitude and random orientation in the center of mass of the ligand. So at time zero, here we have our protein-ligand complex, we have our ligands in pink, and then we apply a force in the center of mass of the ligands. So after time delta t, we check the displacements of the ligands. If this displacement delta x was larger than a threshold that we established, then we keep this force orientation. But if the displacement was smaller than the threshold, then we change the orientation of the force randomly. And we keep doing during our simulation these steps of propagating the motion of the system and checking the displacements of the ligands until we have the phenomena that we want to achieve, that in this case is protein-ligand unbinding. So now something important is that we are applying a force over the system, so we no longer have absolute timescales or absolute kinetic rates, because we're applying a force and we are not making corrections for this, but we still have what we can have for a group of ligands if we are using the same force is the relative kinetic rates. And these relative kinetic rates should be able to provide the same ranking as the experimental kinetic rates. Okay. And then, okay, now going to the results. So for simulations, first thing we did was to do simulations for all, for the wild type and the 10 mutants that I showed you in the table in the introduction. and every point here corresponds to the average of 75 independent simulations or independent trajectories. Here we have the experimental residence times, and here we have the computed residence times for CO dissociation using a force of 1 kilocal per mole per angstrom. And here we have the catalytic sites of the hydrogenase. CO here is represented in green and red. So this is just one example of an unbinding pathway for CEO. So CEO starts here, travel around, and then goes outside the protein. And here we have we have a Pearson correlation coefficient, sorry, of 0.79, and a Spearman correlation coefficient of 0.75. The maximum values they can have is 1. So this shows to us that Taurangi does a reasonable job in ranking the relative residence times for this system. So once we see that our simulations reproduce the experiments, then we go for further data analysis. So remember that we wanted to test those two bottlenecks proposed in the literature and see if they were really determining the residence times for CO dissociation. So here we compare the experimental residence time with the first bottleneck, the distance between the terminal residues in position 74 and 122. And we have a strong and negative correlation here. So this means that this bottleneck is an important determinant of this residence time. So for instance, when we have a short distance between these two residues, this means we also have a long residence time. And now for the second bottleneck, we do see some correlation here. We have an R value of 0.58, but we have a positive value here, meaning that here, the distance between residues 74 and 476 that you can see here, this distance is not acting as a bottleneck for CO dissociation. So for instance, if we look at this data point, when we have a long distance between these two residues, we also have a long residence time. So this the distance between these two residues is not really acting as a bottleneck, as is the case for these distances here. And then finally, talking about the tunnels, which was the basis for the development of our machine learning model later. So first, we analyzed the crystal structure of nickel ion hydrogenase using K-Verdoc. And with this analysis, we identified 9 independent tunnels that are represented in different colors here, T1, T2, up to T9. And then back to our simulations, we analyzed our simulations using Aqueduct to match, and we did a matching between the pathways in Aqueduct with these tunnels identified by K-Verdoc. And here in all these graphs, you can see here we have the tunnels, and here we have the probability of finding these tunnels in all the 75 independent simulations for each data point. So one of the questions we had was, do the mutations in hydrogenase change the unbinding pathway? And we can see here in these graphs that not really. So in the wild type or in different mutations, we can see that pathways T1 and T2, these pathways here, T1 and T2, are the preferred pathways for unbinding in all mutants. And yeah, so that's the first thing. But what I find very exciting about this work is looking at the pathway utilization for different gas molecules. So here we just have the pathways for CO dissociation. So now we are looking only at the pathways in the wild type hydrogenase for CO, O2, and H2 dissociation. So remember that CO and O2 are inhibitors, so we want to prevent them from accessing the binding site, and H2 is a substrate. So we only want to see H2 reaching the catalytic site of this enzyme. And here again, when we look at the pathway probabilities, we see that the primary pathways, the most probable pathways, are still T1 and T2 for the different gas molecules. So CO, O2, and also H2. But what I find exciting is that when we look at the secondary pathways are different for the different gas molecules. And in particular, for H2, we see that we have a higher chance of utilization of alternative pathways like T4 and T9, which are mostly utilized by H2, but not by O2 or CEO. So based on this different utilization of secondary pathways, we propose in the paper that we can we can introduce mutations in the hydrogenase to first of all block pathways T1 and T2, and second, open up the pathways T4 and T9 that are used mostly by the substrate only. And now, finally going to the machine learning part, that is, it's not officially published yet, but it's available on ChemArchive and also on GitHub. So one of the challenges we had in this work and the motivation we had to build our machine learning model. So we have here, again, each point here corresponds to 75 trajectories, and we have a total of 11 points or 11 enzyme forms, meaning that we had to analyze 825 trajectories. And this was very challenging, and using Caver and Aqueduct was helpful for us, but this posed us a bit of challenging, and it required for us a bit of visual inspection to match the pathways in different mutants. So what we wanted to do was to streamline our analysis and make it faster and more convenient, and also an analysis that would require from us less visual inspection. So that's why we wanted to propose PathInHydro, which is a set of machine learning models to identify unbinding pathways and unbinding trajectories in an automated way. And this, again, this is available on ChemArchive if you want to see. The algorithm we use here is random forest, which is basically an ensemble of decision trees. I guess you must be familiar, but I can make a quick explanation here. So this, for instance, is a simple example of a decision tree to predict solubility using two features, the log P value and the molecular weight. So a decision tree is made out of these nodes, and then in the end, with all the questions we ask, we make a decision for our data point. So what we have basically here is a hierarchy of questions that we ask about our data point. So first question we have here is whether the log P of our specific molecule is smaller or larger than 3.11. And then if it's smaller, we go here. If it's larger, we go here. And here we have another question. Is the molecular weight smaller or larger than this value, 268 grams per liter? And if it's smaller, it goes here. And then if it's larger, it goes here. And then with this, we make a prediction for the specific data point. And then again, we use random forest, which is just not only one decision tree, but it's a group of decision trees that helps to overcome the trends of decision trees in overfitting. And this is our data set. So we proposed two groups of models here, one model for binary classification and one model for multi-class classification. And I presented the first part to you, the trajectories for CEO H2102 unbinding, because those trajectories were our data set for the machine learning model. So for our training set, we used the trajectories of CO dissociation from the nickel ion hydrogenase of the Desulfovibrio fructosovorans. And as a test set to test how generalizable our model would be, we tested not only CO, but we also tested two other gas molecules, H2 and O2, and we also tested CO dissociation from another hydrogenase, the hydrogenase from this mega Desulfovibrio gigas, which is another hydrogenase, another organism. And its features, we use the fraction of time that we use the fraction of time of the ligand residue contact during the simulation. And here we have, for instance, the data set for the binary model. So our primary pathways here were the pathways with highest probability. So pathways T1 and T2, which are here in green and cyan, together with pathway T7. And we grouped these pathways together, first of all, because T1 and T2, they have high probability, and second, because T1, T2, and T7, they are structurally related. So the first half of the pathways is very similar. And the secondary pathways are all the other pathways that do not involve T1, T2, and T7. So all the other 6 pathways that we have here in this figure. And first problem you can see in this data set is that we have a huge data imbalance, meaning that we have much more cases of primary pathways than secondary pathways. To overcome this, we used oversampling to increase the amount of data points for the secondary pathways for the less represented class. And this again is the data set for the binary model. So here is what we use for the training set, only data for CEO. And here we have the test sets for CO, H2, O2, and the other hydrogenase. And we also had a multi-class model. So here we are basically when we look at the trajectory, we are making a classification for this trajectory for 1 out of 9 different pathways. So here we represent basically all the 9 pathways that we can see in the crystal structure and in the trajectories. And this is way more challenging because we have now 9 pathways. So we have 1 correct answer and 8 different ways that we can be wrong here for a specific data point. And we have, again, the problem of data imbalance because the most probable pathways, again, are T1 and T2, while we have very few instances of the other pathways. And we, again, use oversampling for this case as well. And for the metrics, we used statistical metrics that were appropriate for imbalanced data sets. So we looked at the balanced accuracy, which is basically the average accuracy considering all classes. And we also use the Matthews correlation coefficient. So both metrics can have a maximum value of 1. Okay, and this is the main table we have in the paper. So this is the performance of the binary model, and this is the performance of the multi-class model. So first thing we did was actually to establish a baseline model. We had this pathway classification for hydrogenases was never done before in the literature. And then what we did is to establish a very simple baseline model, meaning that we had a model where we attributed all the data points to the most abundant class. In this case, the primary pathways, and in this case, the tunnel T1. And these are the statistics we had for the baseline model. So this is what we achieved using the training set, which are metrics close of 1 or very close to 1. And here we can see the results for the test set for the different gas molecules, CO, H2, and O2, and also for a different hydrogenase. So I find very exciting to see these metrics for the different test sets, because this means that the model is generalizable and it's applicable not only for CEO, but also for other different gas molecules, because the metrics don't drop too much in comparison to the binary, in comparison to the training set, sorry. And the other thing, if we compare the metrics, we see that we have a slightly lower performance for O2, especially when we look at the Matthews correlation coefficient. So O2 has a slightly worse performance here and also here. And this is something that we found curious, and our ideas here is that maybe this happened because O2 has a larger size in comparison to the other molecules, or maybe because O2 is making different interactions with the enzyme in comparison to CO, for instance, which was used in the training set. And just to complement what we discussed, these are the rock curves for the test sets of the binary model. And we can see here that we have the area under the rock curve, very high values, which are close to 1, the best value that we can have. And here, the other way we can look at classification problems or to look at the performance of the models is looking at confusion matrices. And here we are looking at confusion matrices for CO and O2 test sets for the multi-class model. And we can see that many of the misassignments we have for the multi-class model, they are internal misassignments, meaning that trajectories that belong to one primary pathway are assigned to another primary pathway. So remember, for instance, that T1, T2, and T7 were grouped together in the binary model. And what we see here for misassignments, so for instance, when we have T1, we have misassignments of T1 as T2 or T7. Which were basically the other 2 similar pathways. And the same happens here. Here we have T1, and T1 is very often misassigned as T2 probably because these pathways are structurally related and share similar features. And that's all I wanted to tell you today. So first, I hope I convinced you that computational methods can be used not only to predict kinetic rates, but also to provide mechanistic insights of protein-ligand binding and unbinding. For the first part, I showed you that different gas molecules can use different secondary pathways for unbinding, which offers us an opportunity to design nickel-ion hydrogenases, which are inhibitor resistant. And for the second part, I showed you our set of machine learning models, PathInHydro, and the inspiration for this was to facilitate the identification of unbinding pathways in simulations of nickel-ion hydrogenases. And something else I want to share. So I'm also an editor of the Journal of Chemical Information and Modeling. You can see my name here. It's a very diverse journal, and we are very much interested in having more papers in machine learning. So please send your machine learning papers to us. And the other thing, JCIM has a lot of special issues going on, something that may be of your interest. We have this special issue with deadline in January 2025 about chemical compound space exploration with multi-scale high-throughput screening and machine learning. And the other thing that is currently happening is that JCIM has an open call for new members of the early career board. And the early career board is mostly made by postdoctoral researchers and assistant professors. So if you are in this career stage, please apply for this open call. I can send you the link later if you're interested on this. And that's it. And the work I presented to you today, this work was mostly made by my PhD student, Farzin, with a lot of help also from my postdoc, Iris Guo. And yeah, and I also want to thank all the funding that made this work possible, especially the Cluster of Excellence here in Germany, UniSysCat. And that's it. Thank you.
Wow. Thank you, Dr. Herein. This is interesting. Uh, First, members, if anyone has a question, you just unmute yourself and pose the question. In the meantime, thank you very much, Doctor. You've talked about the last thing I want to start with is the JCM journal. Actually, I was looking for its template, the LaTeX template for JCM.
Okay. I think there's a general LaTeX template for ACS journals in general, and then you have to adjust it a little bit for JCIM, but there is a template for sure.
Okay. I got confused. I saw quite a number of them, and I was not sure which ones, which so I decided to first type my paper in general LaTeX, then maybe when it is ready. But since now I have you, I think that won't be a problem. For me, my first question is about the machine learning part.
Mhm.
And I've been wondering what can be said about the data representation of to represent these pathways, like in terms of sort of visualization, but also, sorry, but also in a way of understanding or looking at the data, how was the data presented, and then at the stage of loading it into the ML models, what can be said about the data representation?
Okay, the data representation. So what we use as features. So first of all, maybe I didn't emphasize this enough. This was a supervised model. So we know what the to which pathway the trajectory belongs, and we wanted to train a machine learning model to recognize this. And the way we represented this, we basically looked at the protein-ligand interactions during the simulation. We basically looked at the let me simplify this. So we basically quantified the amount of time that the residue was interacting, a residue in the protein was interacting with the ligand during the simulation. And this is how we represented the trajectories for the training. These were basically the features that we used. Is it clear?
Okay. Still, it is still abstract in the sense that as someone would wonder, okay, these pathways and the trajectories, do they have some sort of numerical representation that we
The
can?
representation of the pathways. Okay, that's a good question. So we have these visual representations from Caver, right? Ah, oh my God. So
Hmm.
yeah, these are representations from Caver by looking only at the static crystal structure. So what we have from Aqueduct does a clustering based on the positions of the ligands during the simulation. And they used as representation, let's say, I think if you check the support information of this paper at JCIM, I have some figures there of how this is represented, but they basically from the coordinates of the ligands unbinding during the simulation, they can generate some lines for visualization, let's put it this way. But yeah, that's kind of a challenging, let's say, but in principle, we can have the representation as coordinates for the ligand position from the simulations.
Okay.
I hope it's more clear now.
Yeah, at least it is more clear. And just wondering, we with some sort of basic uh, uh, visualization software like PyMol, how far can I achieve?
Aqueduct, sorry, go ahead.
Uh, can I achieve this level of data, like sort of visualization with PyMol when it comes to these uh, proteins, the
Yeah,
enzymes? Sorry.
so ah,
Or
so
there are other tools that.
all these figures they were mostly generated using PyMol. So CaverDoc is a plugin available for PyMol, so you can download it and install it in PyMol, let's say, and then but CaverDoc analyzes these tunnels or pathways for crystal structures only. An Aqueduct it analyzes trajectories and it generates an output file that can be visualized with PyMol as well.
Okay.
So the two things can be downloaded for free and be used together with PyMol. That's in fact exactly what we did. We used these two tools in combination with PyMol.
Another itching question for me, Doctor, was about when I see it's sort of a bias. Um, when I see people opting for machine learning models, and I want to think about these pathways as a complex sort of pattern, intertwined, like unpredictable in form. I could be wrong about that, but that's what I'm getting from the presentation. Now, my question is, wouldn't it be better, or wouldn't deep learning models like achieve greater performance and prediction, like graph neural networks in that case? I don't know.
The reason, yeah, I agree that for some complex tasks, like, yeah, a lot of things, neural networks or graph neural networks could be better. For this data set particularly, I was not so happy of applying neural networks here because the data set is not so large. So we have for CEO, for instance, 800 trajectories, and we still split this number of trajectories in training and test set, meaning that the number for training is even smaller than 800 data points. And this is not a lot in comparison to the number of parameters that we have to, or weights that we have to adjust in a neural network, in a deep neural network. So that's why we, for this particular project, we decided to not go for neural networks, or it's
Okay.
more complicated.
I have read this paper, and I still have some interesting, intriguing questions.
Ask me anything. Yeah.
Somewhere you talked about the residence time. Was it residence time, contact time? But I think it's about gas molecules and enzymes, like that sort of engagement. And uh, whenever I see this, I have interested myself in proteins, but also in their geometrical representation, topological structures, so to say. And where sort of these metrics, like the angles of sort of interaction, or the topological representations of these proteins, sort of if we are to decide to look at them, would that change the effect of these gases on these enzymes? And all enzymes structured in a different way would maybe uh, resist, or would have a better survival on certain gases?
Yeah, so our whole idea here was to potentially see if we could introduce mutations here, for instance, in the vicinity of the catalytic site, or other mutations that could potentially change these pathways for binding and unbinding. And this is yeah, somehow related to the topology in the sense that we change the tunnels or the space available is inside the enzymes.
Okay. Um, Lastly, I think for me is you've talked that you've said the data was quite small, and I think not quite, but maybe was not enough for deep learning models. And um, what can be said about the number of features, or sort of like the dimensionality of the features that were used for machine learning in the random forest? And the point where I want to ride at or arrive at is about dimensionality reduction. Was there, because I felt like maybe the features were too much, or they were not overwhelming. So in brief, was there some sort of dimensionality reduction done due to the number of features?
That's a good point. So here we had a lot of features because this enzyme is quite big. I think it has around 500 residues, or meaning, so each residue will generate a feature. So 500 features, let's say, and I think my PhD student Farzin tested random forests with a smaller number of features, and he didn't see big changes in his model. And yeah, I agree that if we have too many features, sometimes we can introduce a lot of noise to the model that and then the model can be less effective. But at least what we tested for this particular model is that doing dimensionality reduction using less features didn't help us very much.
There is a question here about noisy trajectories in the different tunnels. How was it addressed?
What was the question? I don't know if I understand the question.
I'm asking about noisy trajectories in where the gas molecules were probably visiting different tunnels.
I see. Yes. Okay, that's something we discussed in the paper as well. Okay, so yeah, what we did, so this is something else. So the simulations, in principle, what we want to see is a very clear unbinding pathway from A to B, right? But what happens is that sometimes the gas molecules doing before coming out, and this introduces noise as well. And mm, and then what we did for this particular model to reduce the amount of noise we had in the trajectories was to cut the part of the trajectory that was equivalent to the bound state when the ligand was close to the catalytic site, and focus more on the pathway of the ligands after it left this main bottleneck that we identified. So, and focusing only on this part and not on the whole simulation helped us a lot to have meaningful models. So in the beginning, when we started this project, we wanted to have features looking at the whole trajectory, and this led to poor models. So we had much less predictive models. And then when we decided to focus on the part of the trajectory after the ligand passed the bottleneck, then apparently we had much more information because the models became much, much better.
Okay. Doctor, I think the last one for me that I can't go without asking is, is there a future extension of this work? Like, what
Yes.
would be the
So
possibilities?
I think what would be very nice, so what we're trying to do right now, so right now, this PathInHydro that we are proposing, it's applicable, first of all, only for nickel ion hydrogenases. And second, this is a supervised model, so it will only be able to identify the 9 pathways that we had in our training set. So if there are new pathways, for instance, we will not be able to identify new pathways because the model was not trained to identify new pathways. So to overcome these problems like restriction to one enzyme and inability to identify new pathways, we are now looking at unsupervised methods to identify pathways in our trajectories. So we are basically looking at clustering algorithms that can take into consideration the time the different protein ligand contacts that we have along the trajectory. And this makes things a bit difficult because we have to align and compare trajectories that have different time lengths. But yeah, we are right now looking at ways of aligning the trajectories and unsupervised learning, because then we would be able to identify new trajectories, new unbinding pathways if they eventually appear in our simulations. And these methods would also be applicable if we make it general enough. This would be useful for any enzyme or any protein, not only hydrogenases.
Okay. Thank you very much, Dr. Herein. This has been fantastic, and taking us through molecular dynamics and then to the machine learning. Unless anyone of the members has a question, I want to thank Dr. Herein for the presentation once again. And yeah, this has been great, and thank you very much. The team here is I don't know, people don't want to turn on their cameras. I would have turned on mine, but the webcam is down. We have Brighton here. Brighton is a PhD student in UT, Texas. We co-organize these talks together. And we have various members. Some I think have not turned up for today, but maybe they will watch the recording. Thank you very much, Doctor, for this.
Okay, thank you for the invitation. Yeah, and if you want to know more about